Okay, can you guys hear me? Yeah, you can. Someone should say that they can. Okay, good. Okay. Yeah, this is gonna be a crazy rabbit hole, random content. I went through this paper for about 6 years. Um, I've had thousands of email correspondences with Maxim, the lead author on this. And the reason that I like this so much is because it's not relying on these pictures or people are giving us like these images or aliens are real because we see UFOs and all this other stuff. What this is like a self-proving type. This self-proves itself. So if you understand it, it loops back and supports it on itself, and that's why I really like it. Um, it's going to take a little bit of work and understanding of the science involved. Um, but if you just be patient with me, guys, this is, as I said, this has been very intimidating to attempt to present to people because of the complexity of it for about 6 years. Okay, you have a PhD in biology, Andres. Okay, thank you. Thank you. All right. Where am I going to start off at? Either paint or just a notepad. So there are two things people get confused a lot: DNA and the genetic code. What is DNA? DNA is like, okay, first off, the genetic code is the language of DNA, like the alphabet for it. DNA is like the book written with it, like that forms an organism. This is a very important thing that's to distinguish here. So DNA, what are the three? It's A, C. I'm gonna have to look up a few things, by the way, because I have to refresh myself a little bit. What are the nucleotides? Okay. There are 4 nucleotides that are a combination of these, make up strands of DNA. So it can be like A, T, G, C, 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 G, T, A, C, A, T, G, all this. And it can get a little bit more complicated, but that's DNA. That's the written book. The C is for cysteine or cytosine, and then guanine, thymine, and adenine. Yes, there's also ATGC. These are going to be. This is really important. Okay, now. Well, we'd have to look at your cat mushroom. Now, let's show you what the genetic code is. Hopefully there's a nice, pretty chart here. Everyone liked it. Okay. everyone likes to make their charts of what the genetic code looks like differently. But essentially, what it means is there are, what is it? Okay, that's a good one. This is a good one, kind of. So, yeah, show it to your genetic engineering professor. professor. So, the genetic code is, what is it? There's 20 different amino acids, and I am going to simplifying this like a little bit. And each of those 20 different amino acids, yes, amino acids, correspond, it might be 21 or something, correspond to a representation of 3 letters of the genetic, of a string of DNA. Well, the woman on Mars, that we're going to get to that. So let's get codon representations. Basically here. Also, sometimes they use different letters for certain things. I think U means it depends upon if okay, it depends upon if you're looking at RNA or DNA. It doesn't matter here. So just assume that the U is representative of T. So we got these amino acids. Let me get a list of them. It's ChatGPT time. These are all the amino acids that each 3-digit, and this each 3-letter combination corresponds to something called a codon. And each of these, there's 64 combinations because it's 4 times 4 times 4. And there are 20 amino acids, so there are some redundancies. We'll see that in a second. And so it's the ribosomes in your cells that are like, hey, okay, we see this letter, like this sequence, this 3-letter sequence. Okay, that corresponds to alanine. Print that. And then they see the next thing. Okay, print that. And they make these chains of amino acids, which are called proteins. And depending upon the various like chemical attributes, like hydrophobia, or the ability to like or want to like absorb water, or all sorts of different things. The combination of all these amino acids makes all the different cells in your like body. That's what we're made of. Okay. Yes. To Andres, they're the most common bases used to build the genetic, but they're not the only ones. Yes, if you get like, you can get deeper into this, and like then there's like long things. There's like stop and start sequences, but the point here is to kind of keep this in a simple way. Richard, it would be a binary code, but I guess with a radix of 4. Not a radix, but whatever. So, okay, the very, very, very important, this is the most important thing here. Let's say life starts out, I mean, okay, first off, we don't know if life started out via something called abiogenesis, which is the creation of life from without life. And I hope you guys can see my screen. Yeah. Well, that's had to have happened, but we don't know if this happened on Earth. If abiogenesis occurred on Earth, or if abiogenesis, the initial creation of life, was created somewhere else and got spread here by like special bacteria that were in cryptobiosis and stuff. That's called panspermia. And I lean towards panspermia happening because like the fact that life would just form, like it seems like such a rarity or such a crazy consequence for just elements and stuff or these amino acids and this information to just form. In a way, I personally view abiogenesis as a sacred event. Scientists have never been able to recreate it in a lab. Sometimes there's a lot of theories about it, but it kind of makes more sense to me that panspermia happened, especially given the fact that there's a lot of bacteria and other microorganisms that can go into cryptobiotic states and likely survive star formation. Yes, asteroids contain the building blocks for life, Jimmy Johnson, but the magic here is not the fact that they have the amino acids, it's the organization of it. It's the information. Yeah, so everything, yes, at the end of the day, abiogenesis had to happen somewhere. Yes, and we can argue about whether or not panspermia happened, or people will say you're just pushing the argument back. However, this paper is not. We're not trying to argue that. We're just looking at something else. We're looking for some sort of signal. Thank you, Razorback. Okay, so. So there's an upcoming mission, I forgot the name of it, that's going to one of Saturn's moons, I believe, or maybe 2 of them, where it's likely that there could be life there due to the conditions present on that planet. It would foster life, or there might be, I think they said like methane outgases or something. And if we find... This is a very big thing. If we find like any life or like some trace of life on another like planet or something like that, then, well, I guess either you can't rule out abiogenesis again, but we've never witnessed abiogenesis happening. Basically what this means that life is likely common in the universe. Well, it's common in the universe. If there's no life in the places where we think it is, then that means we're really alone. Also, if you find life in somewhere else in our solar system, I think that's a very strong argument for panspermia occurring and coming to our planet during star formation, where there's a giant disk and all the planets are going around and stuff like that. And it's actually very easy to like seed star forming nebulas if you just send out a bunch of solar sails with like bacteria. So, okay. I think we've gotten this over with. We got this thing. Now I'm gonna start kind of moving towards the paper and the way Maxim, the lead author, wrote this. I've been telling him for so long that I'm gonna do this. Yes, Andres, exactly. So those signals, actually, okay. So the genetic code did not change. It cannot change, like the alphabet of how DNA is passing on information from evolution to evolution cannot change because you would lose information. So the genetic code is in essence, except for a few outliers like fruit flies and stuff, in essence, the genetic code is a locked language for life on Earth. Like, and this is very, very important. Like, it's been the same since Lucia. Maybe it's like a few slight variations or something, but it's called the canonical genetic code. This is very, very important. So, everyone agrees, everyone understands that the genetic code could not have changed, or because if it did, life would not have the information it needed to evolve from one form to the next, because it would be like trying to communicate information in a different language. Now, think about that. If the genetic code cannot change, and let's say you were like an ancient extraterrestrial civilization somewhere, way off somewhere, and you found that abiogenesis was really truly a very sacred event that occurred. And well, okay, skip that part. Let's say you're an alien civilization and you want to leave some sort of mark or some sort of signal or something that maybe some intelligent species will find in the future. What are your options to leave on a planet to mark the fact that there's you guys were at this planet, besides UFOs and stuff? Like, let's think about this. We're some aliens. We want to make sure. I mean, you know, Earth has been around for 4 billion years. It's gonna be around for a while. We want to leave something that's gonna stay. So, I'll put out some options. We're aliens. Let's build some big ass pyramids. Okay. That's retarded because those are only going to last for like 4,000 or 5,000 years or something. Let's mess with some DNA somewhere, like hybrids. No, that's going to get washed out eventually. I can't think of any others. Okay, geographic markers. Yeah, like the Earth has changed a lot. Technological, any sort of technological marker, like everything's gonna rust away. Everything's like any technological thing will rust away or die and be thrown out to entropy. So any tech, I mean, like, there's just nothing that there's nothing that can be made that's going to last billions of years on a planet that has gone through like like the moon smashing into it and stuff. Yes, Dave, and this is sort of where we're going, the genetic code. So, we come to the conclusion. What is the one thing that will not change since life began on Earth? Because it cannot change. The genetic code. It's the one thing that isn't going to change because it can't change. Otherwise, life would it just wouldn't work. So if you want to put a message in there, put a message somewhere that lasts for billions of years, you put it in the genetic code. Something big in space could last a lot longer. Not necessarily. For 4 billion years? Crazy thing is biology or DNA's ultimate goal could be. Well, I'm going to try to not go off on too many tangents. I am reading chat, though. But the genetic code is the best place, I hope you all understand why, to put a message to some sort of life form that might eventually evolve out of life. Okay, cool. Moons can go, like a lot of stuff can happen, but life remains. Well, you kind of are a robot, Jimmy Johnson, just an organic one. Okay. No, shit. Corrupt skies. This is where USS Liberty. It doesn't matter because because of the way that you'll see things line up. It doesn't matter what we would assign the letters and stuff like that. Okay. I'll read this abstract like really quick. I don't want to bore you guys, but it's basically what I just said. After it was proposed that life on Earth might descend from seeding by an earlier extraterrestrial civilization motivated to secure and spread life, some authors noted that this alternative offers a testable implication. Microbial seeds could be intentionally supplied with a durable signature that might be found in extant organisms. In particular, that the optimal location for such an artifact is the genetic code, as the least evolving part of cells. However, as the mainstream view goes, this scenario is too speculative and cannot be meaningfully tested because encoding decoding a signature within the genetic code is something ill-defined. Basically, when this was published, it was considered some sort of pseudoscience or numerology. I think nowadays it's a bit more accepted, and a lot of the people that criticize this paper didn't read it. So he basically Maxim here essentially goes through a lot of things that I just said. That he just really substantiates like a lot more farther. He's explaining how if some ancient aliens wanted to leave a mark in the genetic code, what they could have done is modified the genetic code, and then they could send them out on solar sails to nebulas. And nebulas are where some sort of thing will disturb the nebula, and then it'll start part of it will start twisting around, and then that starts forming a star, and then planets like kind of start forming around that star, and so like nebulas are our stellar nurseries. So if you just get some life there that can survive it, and you want to leave a little mark, then you could do it this way. And we can do this nowadays, even with like some solar sails, and we just like if the bacteria or the life can survive in a cryptobiotic state for so long, then like it doesn't even matter how far away the like the nebulas are. Like, it could be going at like 10% the speed of light, which is feasible with a solar sail, and maybe it'll arrive in 100,000 years. Doesn't matter. And there's a motive for them to do this if they believe that life is sacred, which could very well be a thing. I believe in aliens in a lot of ways. Eagles Freak, isn't our DNA changing over time though? Our DNA is changing and evolving, but the language of our DNA is not. Okay. So, we're going to get into a bit of biology. Dun, dun, dun. He talks about how like genomics, like something found inside DNA, like that's not going to find anything. So we get to this part of and this is going to be a very important relationship. The relationship between codons, triplets of I'm going to say nucleosides. And the amino acid they code for. Or you could maybe nucleons. As he says here, useful information might be encoded into how these sets are mapped to each other. Interesting. Now, there is also something very important. It is the definition of what a message is. Nope. Okay, a message, you're going to have to think a little bit deeper here. What is a message? A message is a symbol or something that is mapped in an order. That could be a temporal order or some other order. So, like, let's say we just have all these like codon, we have all these amino acids, and we have all these like we have 64 codons, which map to 20 amino acids. Now, if I were to make up an alphabet of like random like symbols or whatever, you would kind of know it's a message because you're either reading it from one way to the other or not. It's like a mapping from one symbols are mapped from 1 to 2, 3, 4, 5, like in an order. There are not quadruplet codons. There's not 2. Okay. Okay, we get to these questions. Specifically, there are 3 immediate questions to address. Is there enough room in the genetic code for an intelligent signature? Does it contradict anything known about the genetic code? Is embedding a signature into the genetic code technically feasible? Well, then we get into information theory. I'm not going to go into that, but if you guys like information theory, it's fun. And there's a lot of theories as to how the genetic code was formed. Those are like, me and Maxim had a lot of arguments about like, oh, it could be this or that. The prevailing theory is that these these assignments of codons to amino acids was called the frozen accident. So it just randomly happened. And okay, so what we've got to do here with our amino acids is we have to figure out a way to order them that would be universally like recognizable, like to an alien or whatever. Okay. So we have to, because like the aliens or the extraterrestrials are not going to name the amino acids that these are coded for, or whatever they mean, with the same alphabet, etc., like we are. So what we're trying to extract here is a universal ordering of these amino acids, because if we obtain an ordering, then that could reveal a message. This is getting a little complex. We don't know what the message is yet, LeBron. We're just looking to see if we can. What we're looking for is a way to order all of these amino acids in a universal way to where there are no duplicates that any intelligent species could recognize. So we have some options here. We could look at, he has a chart here for all of these. We could look at like the number of double bonds in the molecule, and then he lists all these. Okay, all these have like 1, 1, 2, 2. What we're looking for is distinct numbers for each of these. I don't need to use ChatGPT to order them. This is very apparent to me, actually. The second column here is the number of hydrogen atoms in the molecule. So he lists all those for each of the amino acids. They're just kind of all over the place. The number of all atoms in a molecule right here. And remember, these have to be... Yes, Eagles Freak. We're attempting to see if we can order these in a way that makes it such that there is an ordering, and then to see if there's something that arises after that. Okay. Atomic numbers right here. They're all over the place for these amino acids. Or no, the atomic number is Z right here. once again. Now, the number of nucleons. What are you confused about, LeBron? I'm probably going too fast. Or if anyone's confused, let me know, because I could see how this is getting confusing. And this is complex, so like if you're confused over what the message is, we haven't arrived at the message yet. We're just trying to kind of deduce a way a message could be like mapped. Yeah. What we need is a way to have an ordering of these amino acids that we're then gonna like put on a chart and see how that lines up with the codons that they represent. Okay, Andres. So you get what's going on, Andres. The geometry would be hard to quantify, but nucleon number, as you can see here, is the most, I'm not going to use the word Shannon entropy. There aren't like many duplicates if you use this number, and it is a universally recognized, like any intelligent species would know how many nucleons that each of these amino acids have. So it doesn't matter what our culture says or how they list them or whatever. And so that means that we're going to order these amino acids by their number of nucleons, because that contains the most possible information. Oh, the pattern, we haven't gotten to the pattern yet, but trust me, we'll see it. I'm going to go pee real quick. Just give me a sec. Okay. Buddy, yeah, it sounds like you've read ahead. USS Liberty, yes, it could. So here we're going to start kind of mapping the... This is going to be an important page right here. If you don't know, these are... so he has listed... Or this is the usual way that people list the genetic code. These symbols N, R, and Y mean something different, and I will explain what they mean in a second. They're called degenerate groups. So, let's see. So the first thing he does is he actually is organizing them into these things called degenerate groups. What are degenerate groups? I'm probably going to lose some people here. If you see an N represents the possibility of that codon being, so this this GGN, the N could be either A, T, C, or G. It could be any of them. For H, that's 3. Let's see, explain what that means. Okay. H standing for T, C, and A. R is for these things called purines, G and A. And Y is for these things called the pyramids. I'm not pronouncing it right, but it doesn't really matter that much. So the first thing I'm going to go over with you is that a logical way to order the genetic code before we order them via nucleon number is to order them via what is called like a how many different codons can be used to program for this amino acid. And that is called, and so like right here, we have these magical 8 ones. Each of these 8 have 4 ways changing in the in position to anything that can code for the respective amino acid. This is SETI in vivo. So these are all that, and thus this is a degenerate group, much like you guys. Then he organizes it by the second degenerate, like least or lesser degenerate group, H, because this I think it's isoleucine. It's ILE. Can be represented by 3 different combinations of codons. The H can be T, C, or A. And then there's this whole group of signals or amino acids that can be represented by 2 codons changing in the 3rd place, R representing G or A, Y representing T or C. And then over here, it's the final grouping of things that can only be represented by 1 codon. Okay, is everyone kind of understand what I'm explaining here with the degenerate groups? Or are we like just lost? Remember, this took me like 6 months to try to understand. Or does anyone understand? Is chat updating? Okay. Thank you, Twiz. Okay. Now notice that in each of these degenerate groups, degeneration, like these groupings, he has ordered them by their nucleon number. Right? Boom. Boom. And boom. It's eagle free. It's a grouping based upon how many different times an amino acid can be programmed by different codons. So for glycine, right here, it can be GG A, GGT, GGC, GGG. And that's the same for all these, like arginine would be CGA, CGT, CGC, CGG. And so that's 4. And so in the next one, this ILEF, that only has 3 different ways of being programmed. AT, ATT, ATC, and ATA. Okay. Okay, so yes. So in biology, these we call degenerate groups because they can perform. There's like multiple functions for the codons. Yes, Andres, it is exactly. And part of the reason is so, there's less screw ups. Hello, Chief Houdini. It's redundant, but it's also degenerate. Degenerate biology has a very different meaning to what people, I think, usually think it means. It means like it's like multiple function, has multiple functions, kind of. Yeah. Yes. So we're gonna move on to C right here. And we're going to keep these degenerate groups in place. We got this one. We got this one. This is third one. Oh, we got this one with the guys who only have 2 possible codons. And then we have this one. Now, before I get into why he reassigned I'm not going to get into the reassignment right now. There's something very interesting here already. There's something very interesting here. Well, before let me get to D first. He makes a change here. Rumer's transformation is taken into account by opposing unsplit boxes. He moves this one over to combine over here. It's a little bit of a complicated explanation as to why. I'm going to skip it for now, but it creates a special symmetry. And this is the final, one of the final, you could call it codograms. It's one of them. And we're going to look at this right now. So there is something called Rumer's rule, which he talks about. For each codon in one half, there is a corresponding codon in the opposite half related to the first one by interchanging T with G and an A with C. This is like the beginnings of clues of some weird symmetries that start to appear. So I can remove all these lines. Okay. For each codon in one half, there is a corresponding codon in the opposite half related to the first one by an interchanging of T and G and A with C. I remember this tripped me up a few times. When he says half here, he means the upper half. So okay, a T with a G and an A with a C. So, as was predicted, interchanging G and T right here, GG, TT. Oh, well, yeah, once again, exact same thing. Now we have G that translates to T, and then you have C, which goes to A, which is right here. And then you can see how, okay, so we have T here, and so we're going to need to find a G and then an A. Well, there's a G and an A here, and there's a G and an A here. Also, bear in mind that this represents 4 different codon combinations. This represents 2. This represents 2. If we look at the C, we should be able to find two A's. So A here, and then C to A here, C to A here, and then C to A here, and C to A here. Right? And then same thing with all of these. This is one of the first weird things. This isn't even about genes. This is about the genetic code. So the first thing there was Rumer's rule. And that's how you can, for every C in the upper part, there's an A corresponding in the bottom part. For every G in the upper part, there's a T corresponding in the bottom part. And likewise. Now let's take all these away again. And remember, we sorted these by nucleon number, right? We tried to get an ordering. So look at this string right here. We're going to paint through it. AGT-AA, AAA-TGA. It's symmetrical. Now, if we look up here, we have another like kind of different symmetry. It's TAT, which is symmetrical with the other side, TAT, but then it's a different type of symmetry across here. And then if we look at these, they're reverse symmetrical. So that's, well, to me, those were the things that stood out like the most. Remember, we ordered these by codons. Yes, and Andres, so this mapping actually also goes in the reverse direction too. Like this isn't just like a bit of numerology. There's some weird stuff that ends up happening. Okay. Yes. Okay. The resulting representation in Figure 1C is a two-part structure with an even perfectory analysis reveals symmetrical features about this representation, mere symmetry of complementary nucleotides in the first string. As might be easily noticed, the structure of the lower part is close to being symmetrical as well. In fact, even in traditional tabular or circular representations of the code, one might readily see that the canonical mapping is almost symmetrical in its degeneracy pattern. There are only two codon boxes, those beginning with TG and AT that are split unequally. This explains why he moved one of the things, but I'm not going to go into that. What he's actually doing here, let me see if I can digest this again. What he's explaining is that there's like a key, and up here, the minimum action to restore like this key is to move over the TGA over here, and he combines it with something or cysteine or something like that. And there's a lot of like references to the movement of a single key unlocking the code. Okay. Now, the final representation of here reveals a bunch of attributes. At the bottom level is Rumer's transformation. All strings of the codogram reveal non-trivial symmetries at the level of the nucleotide symbols. And also with the syntactic symmetry of the start and stop symbols. Okay. Okay, also let's look inside highly conserved sequences, genes we've inherited from Lusa. I mean, we can, but at the same time, this is really about the genetic code. It was published in 2017. And the link to it, I can give you the link to it somewhere. What happened was a lot of biologists didn't like the interpretation of this. And there's a lot more like hidden symmetries and weird stuff. And even the, like, there's a bunch of redundant numbers with the radix of like 7. And this part gets a little... okay, so this part got a little confusing to me before, but I'm just going to try and like go through it on this stream. He has some videos for it, but let me see if I can bring up his videos. How many letters make up a gene? It could be like various amounts. Chifudini, it's a code within something called the genetic code, which is universal to all life. And we're going to look at these videos now. Okay. Okay. Let's see if this can make some sense. Okay, so that's what we had before. And now he's showing what I showed before. And the other one. And you select a codon is mapped under this inversion to a codon in the opposite half of the codogram. Like that. Fuck off. What he's saying here is exactly, yeah. And now he's revealing more symmetries. Now remember, we sorted these by degeneracy groups and then by the nucleon number. And this is what happened. Full mirror symmetry. Yeah, I understand aperture. I had to learn a lot to really get this. I also tried to make my own genetic code with a lot of permutations, and I failed. This is like a really weird setup. Yes, Twiz. So there is a hidden message in the code. It is revealed via symmetries and a few other things once we get to the next mapping. And those symmetries... Okay, hold on. We're getting crazy here. Those symmetries and the way that things point towards... I'm skipping ahead here. Things will end up pointing towards the idea and not the number of 0. 0 is not a number. It is a symbol, and it was developed from culture and even like at different times around the world by different cultures. 0 doesn't exist in nature. So what this ends up doing is pointing towards this symbol of 0 at the end. If you, this, I mean, remember, this is a lot, as I was saying. And 0 is a code of culture, meaning it's an attempt to try to convey this symbol of 0 to us and say to whomever is reading this or looking at this, we were here. And the stream is also, and furthermore, it's basically saying that there were some there's some aliens that were like, I don't know, maybe they started 4 billion years into the universe, life started, and then they started spreading around life throughout the universe, which would be very easy. And they wanted to leave a little like trace of saying that, yeah, by the way, you had some fathers a long time ago. Well, Andreas, don't you think that the most highly conserved thing would be the genetic code? So there's a bunch of weird symmetries that show up when you align things with the degenerate groups and then with the nucleon numbers. I mean, we could look at, yeah. Okay. Now we're mapping the amino acids to their side chains. So for you guys that understand chemistry here, the side chains here, it's like these are the amount of, I believe, the number of nucleons in the side chain for each of these things. And then the number of nucleons for, maybe this is like the base. I don't know what this is called. And what he's going to do is sum them all together. And you'll find that they all equal this, they both equal the same number. Andres, did you see that? Because I know you know this. Remember, this is also universal because Like we're just like any species could do this. Any intelligent species could add things together and see that regardless of how their number system works, and see that the what's up here matches what's down here in number. Okay. Notice that whomever's trying to convey this is assuming that our number system likely has a low radix, or is trying to do things in certain radix, I believe. Okay. I didn't think that I would go this far into this, but let's go from this as well. I'm going to play this. I think I've overwhelmed people, but this is a trial run. The radix isn't universal, which we'll get into in a second. Okay. Here he gets into the activation key, which is this proline thing. So he's talking about, you line all these up, and he's looking at the chemistry, and there's this key to kind of unlock all these symmetries, and it ends up being, it looks like moving something from a side chain to proline. And then if you do this in other places, you'll see a bunch of more things unlock. So shifting it, the key, because if it doesn't shift, it's a denial and doesn't work. The way he kind of phrased this was that, I know what you mean. But I have to go into what he said before in the paper to kind of really explain it. Then again, he's showing you in like 80 different ways. But most organisms use the same genetic code. There are some exceptions, like fruit flies or something. Yes, you are right, Andres Jones. Because the radix of our system is 10. Or 9. We're almost done. Smash that like button. So that's it. I mean, you saw what I saw. I have always wanted to attempt to explain this in a much more clear way, but I think you can see the complexity of it. Especially when you're just throwing a bunch of information at once. But I do want to get to the conclusion. And there's like a lot of testing that has been that this guy, I tried to prove this guy wrong for like 2 years, and I failed. But there we go. Yeah, I'll hit them up again, Houdini. If this makes sense to anyone right now, with that, okay. Given such efficiency and universality, it is likely that during technological evolution, civilizations typically converge on using zero-based positional notation, though of course the choice of radix is still arbitrary. Already within human society, several cultures converge independently on using positional numeral systems. With that, zero-based positional notation is still a code of culture, not a phenomenon of nature. In this sense, preferred notation system is a stronger sign of artificiality than something like prime numbers, which are often mentioned in the context of SETI, simply because no natural process capable of generating such numbers is known, not because they're a code of culture. The most straightforward way to demonstrate zero-based positional system with a preferred radix to another preserver is to produce quantities that have homogeneous notation, composed of identical digits. And this is exactly what is observed. And there's a lot more in here, but we get to a conclusion. Basically, we find that not only the structure and attributes in the genetic code is consistent with the initial assumption, but that it makes perfect sense from the SETI perspective as it encodes some of the most prominent universals of culture. The zero-based positional notation encoded in the direction from codons to amino acids, and the notion of zero as a number in its own right, encoded explicitly in the opposite direction from amino acids to codons. This appears sufficient to interpret the message as, to whom it may concern, we were here. Andres Jonas, yeah, Maxim Makukov, he's one of the smartest people I've ever interacted with. Very patient, too. The symmetries are like a way of showing to like any observer that, okay, there's like something going on here that's artificial. What codes? No, it's the genetic code is for all life, except for like a few weird species. Here. Virtually all known life on Earth uses the canonical genetic code, meaning the vast majority of organisms from bacteria to humans utilize the same set of rules to translate DNA into proteins, with only a few minor variations found in certain organelles like mitochondria and some species. So essentially this language that I was describing is in all life. So yeah. The Andres bacteria do not have a different genetic code than ours. So yeah, I've covered this. This was a rough attempt. I do see the sticking points and how things are confusing. I think this really has to be presented in a way that is in chunks and really highlight certain aspects of it. Also, if you don't understand certain things about information theory, it it gets like you get a little bit confused. It's just it is a lot to kind of take in. So I think with that, I'm probably going to end this stream. I'm going to get back more into UAP stuff and stuff like that. Yeah, the whole idea is really interesting, and what gets weird is when you spend 2 years trying to prove it wrong, and then you realize that the author like just keeps being like, no, that it's wrong because like your thoughts are wrong here because of this. The most ancient organisms still use the same genetic code. It doesn't matter what the genome. Yeah, I didn't expect too many views on this live stream, but I did want to do it to see what happened. So I do have some ideas for some next content while we see what happens with updates in the leaker saga. I've been doing some like photo forensics. JNN, there's just, I guess there's so many symmetries, and when you align them in these things in a way that could allow them to contain enough information, then these symmetries appear, and it seems like they appear like something. I mean, they look artificial. And then the thing is, he also maps it the other direction. So there's a lot of kind of weird things going on. At times, the author was accused of like numerology, which is kind of like just finding things where they aren't really there. But if you go through this paper very clearly and see the logic behind it, it kind of ends up being like a symphony. And there are some difficult things to attempt to argue against. It's just the problem is, and also he had this reviewed. It was published in a pretty high-ranking journal. I think it was a Cambridge journal. But it conflicts with so many other people's theories on how the genetic code developed that it was stigmatized. No, this info did not come from the whistleblower. This came from me researching this paper and knowing the author very well for about 6 years. Maybe symmetry is a necessity to life being possible. Yeah, Twiz, no, I understand. I really rushed you guys with this information. So, I tried to really prove this guy wrong in a lot of ways. He does do like a something in, he programmed something in C to test all other potential like variations of the genetic code, and he like can't find anything. And then he goes into all the other theories of how the genetic code formed, the adaptive hypothesis, stereochemical, biosynthetic. Yeah. If you email him, he will respond too. So it seems that with Mr. Immaculate that I said something he did not want me to say on the Night Shift podcast about how he recommended that I should. I'm not going to repeat it. And he felt that that kind of like undermined his credibility, and then he deleted his email. So he may end up emailing me back at some point. I'm not sure. I don't really know what's going to happen with that story. I don't know. I will tell you that I do have Lou Elizondo's real email address, which I might hit up and see what happens. Hyperish. Here, let me put your stuff on the screen, guys. Hyperish, I would like to, in a segment of videos with a lot of animations that really make things make easy sense. And that's why I haven't released this for so long, because it was too, like, I have to get this perfect. I have to get this perfect so people see the light. But today I was just like, okay, I'm just going to, you got to start somewhere. That's my mic. I'm okay. Can you hear me better now? Yeah. JNN, do I think he was legit? I think he was. He demonstrated characteristics of someone who would be in as complex of a political and internal situation among multiple factions who are all trying to get full disclosure, but want to do it differently. Yeah, I'm not sure if I'll hit up Joe Rogan. Andres, that's what Maxim Makukov's dream is for this. Like, he was at a big conference. I remember him telling me this, and like Avi Loeb was there and stuff like that. There is a Discord, Chief Houdini. It works now. And a lot of people just kind of like, there's a lot of ego in academia, and like everyone wants to be the first to discover something or this or that. And I know that like he like Maxim Makukov put a lot of effort into this, and it spans about like four, five, six different domains of science that I had to kind of teach myself from watching YouTube videos. But all the scientists kind of ignored him. What do you guys think about me taking UFO footage and then doing forensic analysis on it? Like, I've gotten pretty good at that. Okay, that doesn't sound like that's interesting to anyone. I may make a video about Mr. Immaculate, but I'm gonna, I think, give him some time. I'm not exactly yeah, so here, I'll show you this website that I've been using for image forensics. okay, for example, this. Why can't I move this? Yeah, Jimmy Johnson. It also had aliasing around the actual thing. One thing I did kind of learn though is so like these images were saved, and you can tell that they were saved. This is the gimbal video, but you can tell that they were saved. Maybe not that one. If there starts to be, it's called JPEG compression. Let me get a good one. So for example, this one. You see how there's a bunch of little like squares here in the noise level analysis? What this is when you resave a file, especially as a JPEG, its compression algorithm uses it like combines all the blocks together, certain blocks together that look the same, and does its best to. It makes these blocks. What you tend to look for if you're looking. So if I were to, so this is a sample image that this guy gives. And he's like, okay, like, this is obviously fake. So one thing you do is, well, you can look at clone detection to see if like the cloning tool is used. But then noise analysis is really important. If you see parts of it that are like whitish, or they don't look like, or they don't match up to the texture that they should match up to, like see how there's a black line there, or there's like some weird things there, then that's an indication that things have been modified. You can also look for things to see if where are we at? You want to see if the reflection of the sun is, or where the sun's shining is like the same, in the same direction. And then there's this other thing, principal component analysis, that it's very complex, but it really reveals if something was like, it doesn't fit into the pixels of the image. And that's like a big one. I'm still learning about how this stuff works. So like the principal component analysis in this image, I mean, you can see how this has been compressed via JPEG, but like there's nothing really weird about this, like the ball in the center. there's nothing that would indicate that it had really been modified. But then again, you can like scan something and get different stuff. I didn't really betray Mr. Immaculate. They were asking for like credentials, like, okay, what are, because that's a key part. And I knew one of the things that he told me sounded like it came from a handbook that would have given him credentials. That's why I told that story. Jimmy Johnson, I believe so. I also took the liberty of printing out all of the stuff myself and then scanning them and then seeing like what happens when you upload them to this tool, and you get a lot of like weird stuff just starts going on. It's very obvious. But I believe these were scans from a printed image. I'm very like confident about that. Well, I guess we'll see you guys later, and we'll see how things go from here. I'd like to go to New Jersey and try and capture a drone, but I can't do that right now. Okay, Andres, my email is in the About page of the YouTube channel if you want to email me. The biggest thing is really just like being able to communicate the BIO-SETI stuff to people. Like I can understand it. It takes me a few hours to get going again on it, but the communication aspect is the hardest part. See you guys.